Biological classification. Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, Kingdom Animalia, viral cells, viroid, and lichens. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system was used till very recently. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. A need was also felt for including, besides gross morphology, other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organisms have hence undergone several changes over time. Not through plant and animal kingdom have been a constant under all different systems. The understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been under differently by different scientists over time. Table 2.1 Characteristics of the five kingdoms. Cell type of Monera. Prokaryotic cell type of Protista, eukaryotic cell type of Fungi, eukaryotic cell type of Plantae, eukaryotic cell type of Animalia, eukaryotic cell wall of Monera, non-cellulosic polysaccharide plus amino acid, cell wall of Protista present in some, cell wall of Fungi present without cellulose, cell wall of Plantae present cellulose, cell wall of Animalia absent. Nuclear membrane of Monera absent, nuclear membrane of Protista present, nuclear membrane of Fungi present, nuclear membrane of Plantae present, nuclear membrane of Animalia present. Body organization of Monera: cellular body organization of Protista, cellular body organization of Fungi, multicellular or loose tissue, body organization of Plantae, tissue or organ, body organization of Animalia, tissue or organ system. Mode of nutrition of Monera: autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic or parasitic. Mode of nutrition of Protista: autotrophic, photosynthetic and heterotrophic. Mode of nutrition of Fungi: heterotrophic, saprophytic or parasitic. Mode of nutrition of Plantae: autotrophic, photosynthetic. Mode of nutrition of Animalia: heterotrophic, holozoic or saprophytic, etc. R.H. Whittaker, 1969, proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. Let us look at this five-kingdom classification to understand the issues and consideration that influenced the classification systems. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, and angiosperm. Under Plantae, the character that unified this whole kingdom was that all the organisms included had a cell wall in their cell. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say, for example, the Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their wall composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in the animal kingdom, which lack the cell wall. It has put together organisms which, in earlier classification, were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification systems which reflect not only the morphological, physiological, and reproductive similarities, but it is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt with separately in chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow, and deep oceans where very few other or life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shapes: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrio, and the spiral spirillum. Though the bacterial structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they do not synthesize their own food but depends on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophils, hot springs, thermoacidophils, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have a chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, is a nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus, canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fusion. Sometimes under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known, can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. 2.2 Kingdom Protista. All single cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protista to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellate, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the Protistian cell body contain a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping cells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomeous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cell. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates, example goniolix, undergo such rapid multiplication that they make the sea appear red. Red toxins released by such large numbers may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, Euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and form fruiting bodies bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air current. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia false feet as in amoeba. Marine forms have silica shell on their surface, some of them such as entamoeba as parasites. Flagellated protozoans, the members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness. Example, trypanosoma. Ciliated protozoans, these are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. Examples, paramecium sporozoans. This includes diverse organisms that have a infectious spores-like stage in their life cycle. The most notorious is Plasmodium malarial parasite, which causes malaria, a disease which has a staggering effect of on human population. 2.3 Kingdom fungi. The fungi constitute unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. When your bread develops a mold or your orange rots, it is called of fungi. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, e.g. yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics, e.g. penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. You have ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated protoplasm. These are called coenocytic hyphae. Others have septa or cross wall in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depends on living plants and animals are called parasites. They can also live as symbiont in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiosperm or zoospore, and sexual reproduction is by oospores, ascospore, and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following three steps: 1. Fusion of protoplasm between two motile or non-motile gametes called plasmogamy. 2. Fusion of two nuclei called karyogamy. 3. Meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately results in haploid cells twin. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage in plus n, that is, two nuclei per cell occur. Such a condition is called a dikaryon and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi from fruiting bodies in which reduction division occur, leading to formation of haploid spores. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood in most moist and damp places, or as obligate parasites on plants. The mycelium is aseptate and coenocytic. Asexual reproduction takes place by zoospores motile or by aplanospore normale. These spores are endogenously produced. In sporangium, a zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or ogamous. Some common examples are mucor, rhizopas, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular. Is penicillium or rarely unicellular is yeast Saccharomyces. They are saprophytic, decomposers, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The sexual spores are conidia produced exogenously on the special mycelium called conidiospores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are aspergillus, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members, like morels and truffles, are edible and are considered delicacies. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushrooms, brackets, fungi of or puffball. They grow in soil, on logs and tree stumps, and in living plant bodies or as parasites, e.g., rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, mushroom, ostelago, smut, and Puccinia rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phase of these fungi are known. When the sexual forms of these fungi are discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colotrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cascuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast, and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plenty includes algae, bryophytes, pteridophytes, gymnosperm, and angiosperm. Life cycle of plenty has two distinct phases: diploid saprophytic and the haploid gametophytic that alternate with each other. The length of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomenon is called alternation of generation. You will study further details of this kingdom in chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fats. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development. Silent features of various phyla are described in chapter 4. 2.6 Viruses, viroid, and lichens. In the five kingdom classification of Whittaker, there is no mention of some acellular organisms like viruses and viroids and lichen. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effect viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? The name virus, that means venom or poisonous fluid, was given by Pasteur D.J. Ivanovsky in 1892. Recognized certain microbes as casual organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filters. M.W. Bazarnik, 1898, demonstrated that the extract of the infected plant of tobacco could cause infections in healthy plants and called the fluid as Contagium vivum fluidum, infectious living fluid. W. M. Stanley in 1935 showed that viruses could be crystallized, and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No virus contains both DNA or RNA. A virus is a nucleoprotein, and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing, and vein clearing, dwarfing, and stunted growth. viroid in 1971. In 1971, Teodena discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobind and the fungal component as mycobind, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas.